I'm an immunologist, and my research focuses on understanding the mechanism that control the chronic inflammation within the CNS. More focus on the innate immune cells, innate immune activity, and these are very important cells because these are not only start the immune response, they can also control and regulate the T cell response or lymphocytes and coming after to induce the pathology. So this is the very interesting question, how we to understand how this inflammation is regulated. With this, we are focusing on understanding the mechanisms, and if we find a pathway that lead to the activation of these cells or the regulation of these cells, we can target that pathway, and that's going to be the huge implication of the several neurological diseases, including neurodegenerative and multiple sclerosis. This is going to be one of the novel approach that we can target not only the cells in the CNS, but also the activation of other cells that involved. And this is more toward the therapeutics that we think we need to develop for progressive MS. As the drug that is currently in the clinic is mainly target the peripheral immune response, and it's not they don't have that much effect on the progressive stage, which is mostly driven by innate immune response in the CNS. With this, my project, or for several years, we're focused on understanding NLRX1. It's an anti-inflammatory innate immune receptor, which is expressed in all cells, mainly localized in mitochondria, and inhibit major inflammatory pathway, including NF-kappa B pathway and the interferon type 1 pathway. And this is NLRX1 can just more complicated picture is suppress the translocation of NF-kappa B subunits to the nuclei, and leads to inhibit production of inflammatory mediators. And with this, it's a molecule that serves as a central regulatory hub for the regulation of inflammatory signals in innate immune cells, including the glial cells in the CNS. NLRX1 express highly with glial cells, and in my project we were looking at how we can dissect the role of NLRX1 in glial cells and how much that would lead to the regulation of neurodegeneration or to stop the neurodegeneration. To answer these questions, we developed several animal models and use those animal models to have different to study NLRX1 in different contexts of inflammatory challenges. For example, one of the very well-known models of multiple sclerosis is experimental autoimmune encephalomyelitis, and I used this model and we induced this model in NLRX1 wild type and knock out the mouse that doesn't have NLRX1, and we focus on anterior visual pathway. It's a unique site that our lab developed expertise to measure the degree of neurodegeneration. The way that it works is we can look at it at the retina and optic nerve. In retina, we can count the number of retinal ganglion cells, and this is where it shows, when that at the end stage of the EAE, we always see retinal ganglion cell loss, and this is the measure of neurodegeneration. And at the optic nerve, we can look at the infiltration of immune cells and activation of glial cells.